The action potential is really going to be beginning where we have the concentration of sodium channels, the voltage-gated sodium channels. And if we have myelin along the length of the axon, then we're going to see that the axon is going to sort of propagate the action potential, leaping from one segment to the other. Okay, we refer to that as being, sorry, sorry, my slides are going. Okay, yeah. So we refer to that as being saltatory conduction. And saltatory conduction means that the action potential in a myelinated neuron does not go to every single segment of the neuron. It's going to be concentrated in the areas where there is no myelin. And myelin is really just fat. We'll talk about how myelin is formed in the nervous system in our next chapter coming up. But myelin is really just insulation for the neuron. And just like with any other insulation in our electric wires, it helps us speed up the conduction velocity. So having this myelin means that the impulse doesn't have to go to every single segment. It can leap to the areas where there is no myelin. We call these nodes of Ranvier, right? Where there's a concentration of sodium channels, and it can leap in between these strips of myelin. All right. So that is to say, heavily myelinated neurons, like our peripheral nerves, the sciatic nerve, the radial nerve, the median nerve, these nerves are going to conduct their impulses much faster because they're heavily myelinated. Thicker neurons, peripheral neurons, especially that are going to be involved with skeletal muscle movement, are usually heavily myelinated and faster. Alternatively, in unmyelinated neurons, right, such as this, we would see that in an unmyelinated neuron, the action potential is going to hit every single segment. These are usually smaller neurons and are usually more involved with either sensory functions or in some cases autonomic functions. So example here, the length of the neuron, site A, site B, site C, the action potential is going to propagate every single segment, right? It's going to depolarize site A, move to site B, and then because site A will now be in its refractory period, that is going to prevent any retrograde movement, right? Movement backwards of the action potential, keeps the action potential going in one direction. So that is how we propagate the impulse in an unmyelinated neuron, and it would hit every single segment followed by the refractory period so it can move in one direction. So just to compare the difference in the rate of the action potential in unmyelinated versus myelinated neurons. And then here's just again for reference what happens in a myelinated neuron. And you can see the myelin sheaths along the axon. You can see the concentration of those sodium channels and the leaping of the action potential between the nodes of Ranvier. Now, lastly, we want to think about how the speed of conduction differs by the type of neuron. So as the diameter increases, the speed increases, right? This helps to decrease any resistance for the spread of that conduction, the spread of that impulse down the length of the neuron. And so myelination is going to encourage saltatory conduction, which by nature is faster. If we think about our very thin neurons, right, with a very small diameter, these are usually associated with unmyelinated neurons. They usually have a slower speed in terms of their conduction velocity. And this is usually going to be associated with autonomic fibers, neurons that go to the cardiac muscle, to smooth muscle, to glands, and things that control our autonomic functions, okay, like heart rate, blood pressure, etc. Now, that can be contrasted with thicker neurons, thicker diameters. This is usually associated with our muscle, neurons going to muscle, either to innervate the muscle somatically to provide motor function, or sensory neurons that are going to muscle. Okay, not necessarily sensory neurons that are going to towards the nervous system, but sensory neurons that are determining things like muscle position, the state of the muscle, muscle stretch, and those types of neurons associated with muscle are going to be thicker, have a faster velocity, and they'll be usually about 100 meters per second in terms of their conduction velocity. So we can contrast the function of myelin with the overall function of the neuron and the diameter of the neuron, right? We can make a comparison of faster neurons associated with muscle versus slower neurons associated with the autonomic function.